Hello, everyone, and thank you for joining us today for the third webinar in our In Silico series co-hosted by the PETA Science Consortium International and the Institute for In Vitro Sciences. I'm Kyle Martin, toxicologist with the PETA Science Consortium International, and I'll be co-moderating today's webinar along with Christy Sullivan, the Vice President of Education and Outreach at the Institute for In Vitro Sciences. Today, we're finishing our In Silico Toxicology 101 module with the second half of the webinar on applications and case studies in silico toxicology. Alongside great presentations, our speakers have generated resources that we hope will be useful to you as we move through the material. On the Science Consortium website, you can find a glossary of terms that will correspond to the presentations, as well as additional resources to enrich today's discussion and your continued education on the topic. Please note that the slides from today's presentations are already available on the consortium website, and a recording from today's webinar will be posted online at this link shortly after the webinar. All future webinar materials from the series will also be available at this link as the series continues. Next slide, please. Today's presentation will include several opportunities for interaction, primarily in the form of polls. To participate, simply scan the QR code on your phone, and the polls will activate once we reach that point in the presentations. You can also direct your browser to slido.com and enter InSilico 101 after the hash. The QR code will be available again on the relevant slides, but we are going to go ahead and take a moment for you to log on. If you would please scan the QR code on screen, so that way you'll have it up. We'll give it a few seconds to do that. Next slide, please. As we head into the new year, we'll be developing and scheduling new webinars in this series. So please stay tuned for updates on the Science Consortium website, as well as all the listservs that you normally follow. Also, the Science Consortium and IIVS are on LinkedIn, and we'll be posting updates there as well. Next slide, please, Amy. We have 3 speakers today who will be presenting on applications and case studies in silico toxicology part 2. We'll hear from Bridgett Hill, Brad Reisfeld, and Bryant Chambers, each of Inotiv. Because their presentations are intended to flow together, I'm going to introduce them all now. Bridgett Hill is a predictive toxicologist at Inotiv, where she focuses on new approach methodologies as potential complements or alternatives to traditional in vivo methods. She's passionate about understanding and addressing big data challenges, such as inconsistent terminology and reporting, to facilitate data use and interpretation. Some of her work includes high-throughput screening annotation using structured ontologies and understanding the variability in zebrafish developmental toxicity responses. She actively engages in public outreach and training activities and seeks to promote the adoption of NAMs. Dr. Brad Reisfeld is a senior principal predictive toxicologist at Inotiv, where he works on various projects related to computational toxicology and NAMs. He's board certified in toxicology and is a fellow of the Academy of Toxicological Sciences. Prior to coming to Inotiv, he was a professor of chemical and biological engineering and public health at Colorado State University. Dr. Reisfeld was a Fulbright scholar and a visiting professor in Thailand, and later spent a sabbatical year as a visiting scientist at IARC in France and EFSA in Italy. He's published numerous papers related to computational toxicology and co-edited a 2-volume book devoted to the subject. Outside of work, he lives on a farm in Colorado and enjoys raising goats and other critters. Dr. Bryant Chambers is a bioinformatician and toxicologist with extensive experience in both government and industry, specializing in computational toxicology and new approach methodologies. He's worked on complex omics projects, including high-throughput transcriptomics and a spatial single-cell analysis. His postdoctoral research at the US EPA focused on developing and supporting computational toxicology tools and analyses such as high-throughput transcriptomics pathways for chemical hazard screening. Dr. Chambers combines a strong background in bioinformatics with modern artificial intelligence techniques, creating hybrid approaches that integrate biological and multi-omic data, literature mining, and ontology mapping. He's an expert in leveraging AI and large language models to advance next-gen risk assessment tools and automated data extraction and analysis. His current work emphasizes integrating complex multi-pathway data to improve chemical safety assessment. So our webinar for today is scheduled for 1 hour. And at the end, we're going to do a Q&A session at the end of the third presentation. Everyone is muted. You can type your questions into the questions module within Zoom toolbar, not the chat module. Please indicate if your question is directed to a specific speaker. Questions that are added to the chat, we will probably redirect you to put them in the Q&A. Please note that for some of the questions put into the Q&A, they may be answered in a written form and will be moved over a category. Some of the questions will be answered live. We've also enabled closed captions. You can turn this feature on or off according to your preferences using the toolbar at the bottom of your screen. So without any further ado, I'm going to go ahead and hand this over to Bridgett Hill. Thank you.
Hello, everyone. Thank you, Kyle, for that introduction, and thank you to the hosts for the opportunity to be here today. Hopefully, you can see my screen. But I'm Bridgett, and I'm really excited to be kicking off the third webinar in the In Silico Toxicology 101 series. So here is an outline for today. I'm going to go through the learning objectives for today's webinar, just briefly touch on a review of in silico NAMs that were highlighted in depth from the first webinar, and then we are going to dive into case studies that were identified through the peer-reviewed literature to highlight applications of 3 in silico methods. I'm going to walk through the quantitative adverse outcome pathways, and then pass it over to my colleagues to describe molecular modeling and AI, machine learning, and deep learning approaches. And then we are going to wrap up with a Q&A. So the learning objectives for today highlight a variety of items related to in silico methods, such as getting an understanding of what's involved in developing, implementing, applying, and verifying in silico NAMs, describing several ways in which an in silico NAM can be evaluated and validated, summarize the drawbacks and benefits of the usage of in silico NAMs, give examples of the types of data needed to support the development and testing of various types of in silico NAMs, identify the strengths and weaknesses of in silico approaches relative to in vivo methods for the case studies presented, and then lastly, recognize some software that's used in the development and evaluation of these. So there is currently a paradigm shift in toxicology towards integrated approaches to testing and assessment. These include the usage of existing information such as in vivo data, in addition to a variety of newer technologies such as the NAMs highlighted in this green area on the left. These are higher throughput, lower cost, and focus on the 3 R's. And in silico NAMs are included in these NAMs used in these approaches, which are any method that predicts the intrinsic properties or fate of a chemical using algorithms and existing data. 2 key concepts to keep in mind whenever you are developing these in silico methods is that not every method fits every purpose. It's important to consider the context of use, which is the intended purpose of that method, such as use in a regulatory applications, what specific toxicological endpoint is the focus of it, in addition to applicability domain, which describes the chemical or biological space that that model is accurately predicting. So now I am going to start focusing on one specific in silico approach, which is quantitative adverse outcome pathways. So the audience, you may all be familiar with sort of a traditional adverse outcome pathway or an AOP where you have a stressor such as a chemical interacting with a biomolecule, creating this molecular initiating event that leads to subsequent key events across multiple levels of biological organization, ultimately leading to an adverse outcome. How quantitative or QAOPs differ is that they add in quantitative information, specifically using models to describe the relationships between these key events, such as key event relationships. And it can be used to answer a variety of different questions. So how can we predict toxicological outcomes such as the adverse outcome? How can we identify critical points of intervention? And how can we use molecular information to predict these key event relationships and adverse outcomes? So Wanted to ask a question just to gauge some background knowledge. So if you would like to participate, Kyle kind of quickly mentioned that we are doing this through Slido and you can access this pool by scanning the QR code. But the question is, which is an approach that can be applied to QAOP development? And I will give everyone that wants to participate a minute to join and then a minute to answer. Okay, it looks like we're getting stable with the amount of answers. So I'm going to go ahead and move on. Thank you so much for participating. And the answer is all of the above. So all of these approaches can be used in isolation or in combination to develop these QAOPs, and it really depends on the level of detail that's needed to answer the specific question. So here are some examples of the types of information that you will need to develop a QAOP. This includes data related to that molecular initiating event, data, dose response data at those multiple levels of biological organizations, so those quantitative relationships can be established. And this data includes anything from in vitro, in vivo, or computational data such as in silico predictions. And then the outputs include you now have quantitative information to describe these key events. They can be used to predict dose responses for multiple key event relationships, including the adverse outcome. It can help identify critical events for intervention, and the outputs can be used for weight of evidence approaches to understand the pathways. And over here on the right is an information flow, just an example of how some of these QAOPs can be structured and designed. So I'm now going to walk through a case study by Foreign et al., and they use existing data to develop a modular QAOP to describe response relationships between key events for screening and prioritization. And so they take this approach where they establish these sort of puzzle pieces for each of the key event relationships along the AOP, ultimately to build a complete puzzle to be able to predict across the framework and ultimately to the adverse outcome. So the models, they had a general modeling approach and then they applied it to two use cases. One is related to more environmental health, such as decreased fish populations, and the other is related to more human health, so impairments to learning and memory. They both are existing AOPs within the AOP wiki, which you can access through this QR code, excuse me. And if you're not familiar with the AOP wiki, it's a free online platform for AOP sharing. There were two major steps to their methods. Their first step was to gather literature or review the literature, gather data related to the key events in the molecular initiating event, and this included empirical data and in silico predictions, and then use that data to create response relationships to facilitate linkages between those key events and the adverse outcome. And so their general approach for these response relationships is shown in this diagram on the right, where they found an endpoint related to a key event and they plotted it against the endpoint for another key event, modeled it so then they could use these relationships to make predictions. And based on data availability, these key events can be adjacent. So it would be key event 1 and key event 2, or non-adjacent, such as key event 1 to key event 3 or 4. And the software that they used was a benchmark dose software. at the US EPA, and if they didn't have enough data or were they weren't able to model using this software, they used regression mathematical equations in Excel. So the first results that I'm going to walk through is related to the decrease in fish populations. So they were able to gather empirical data for the majority of the key events in the molecular initiating event to be able to model those relationships. There was one instance where they could only find 2 data points, so they ended up using a quadratic equation to find the best fit. But the examples on the right here show some of those dose response relationships that they were able to model using the software. The top one here has a estrogen hormone concentration plotted against an egg yolk precursor protein. And then on the bottom is that same exact egg precursor protein related to another key event. And as you can see, based on how those data related to each other, a different best fit model was selected. They also used in silico predictions to associate a key event to the adverse outcome. So here's just an example of where along that AOP framework they were able to establish these quantitative key event relationships. On the top here is the starting conceptual framework from that existing traditional AOP. And then on the bottom here is where exactly they were able to create those modules to facilitate those relationships between the molecular initiating event and key events. This is a non-adjacent key event relationship. They weren't able to collect enough data to have adjacent key events, but this can show you where the data gaps are along this AOP and where future work is needed. But just to show an example of some of the predictions that you can make, so looking at this specific module on this response relationship, you can make predictions and estimate if, for example, a 45% reduction in one of the key events, such as the plasma estrogen hormone levels, can lead to 53% reduction in that egg yolk precursor protein. And you can continue making those predictions and associations as you move closer to the adverse outcome. Now I'm going to walk through the results for the second use case, which was impairments to learning and memory. So this QOP was a little bit different than the fish populations in that they initially started on a specific compound, an amino acid, due to the complexity and the data gathered for this AOP. There was also some data normalization techniques that needed to happen. This involved different mammalian species, and they ended up ranking responses on a 0 to 1 scale, for example. And then to increase the predictive capacity of this QAOP, they also found some information related to a known neurotoxin so that they could make associations there. And so the dose response for a synthetic amino acid that they used for the majority of the QAOP development is shown here on the top, and then the dose response model for that specific known neurotoxin is shown on the bottom. And as you can see, the shapes of the curves are kind of similar. Different models were used, but the concentrations vastly differ. So here is what their final QAOP looked like for the learning and memory impairments. Once again, non-adjacent key event response relationships were found for this one, also highlighting where the data gaps are that could use future work. One thing that I wanted to highlight is with this one, even though it's a linear AOP and not a network, it had 2 different options for one of the key events. And even though they didn't establish response relationships with these, I just wanted to highlight that the QAOPs and AOPs can vary in their complexity. I did want to highlight how the 2 dose response relationships were used in this example. So as I mentioned on the previous slide, this QAOP focused on a specific compound. And so what they ended up doing to test the predictive capacity or increase the predictive capacity, they found the same endpoint for those two chemicals, and they used it to make predictions for other response relationships later on in the AOP, since it focused on that specific amino acid. So you could associate, what concentration of this neurotoxicant was the equivalent concentration of this amino acid that caused a similar response, and then use that for your predictions later on in the AOP. And then you can also make similar conclusions, such as using these response relationships to say, okay, I'm really interested in what concentration of glutamate in this example would result in 50% neuronal survival for that particular key event relationship. So in summary, hopefully you have a better idea of QAOP development and the information that's needed. Here we showed the creation of a modular approach in building QAOPs using existing information. Some of the strengths of this approach was the demonstration of using this in a pretty wide applicability of building QAOPs in this way of, creating those puzzle pieces that can be fit together. And this was highlighted based on the two different use cases. And while there's now quantitative data still associated to it, it's still pretty chemical agnostic, in which they can be built upon as more information is collected. Some of the weaknesses were the uncertainty was not determined for the best fit curves, and those small data sets for some of the key event relationships can hinder the confidence of the predictions. I did want to note that we are preparing some additional resources that will have other examples of QAOPs and give you an idea of how complex and different these approaches can be. And so I want to thank everyone so much for your attention. As a reminder, if you have any questions, please feel free to put them in the Q&A box. And now I will pass it over to Brad.
And to our audience, Brad Reisfeld was unable to attend today, but he did go through the process of recording his presentation. So our middle presentation will be a recording. Thank you.
Hello, everyone. My name is Brad Reisfeld. I'm a senior principal predictive toxicologist at Inotiv. My sincere thanks to the PETA Science Consortium International and Institute for In Vitro Sciences for sponsoring this webinar series, and for the opportunity for me to speak with you today about a case study in molecular modeling. Let's first do a brief recap of what was presented in webinar 1 regarding molecular modeling. Molecular modeling is a broad field, and for the purposes of applications in toxicology, I like to think of it as consisting of 4 main areas. Those are shown as the leaves of this hierarchy. There's quantum or computational quantum chemistry, molecular mechanics, molecular dynamics, and molecular docking. Our case study today is related to molecular docking, so let's focus on that. As a brief reminder, molecular docking predicts how a small molecule fits into a protein binding site by computationally sampling many possible positions, orientations, and conformations of the molecule. These are called poses. Each pose is evaluated with a scoring function that assesses the goodness of this binding, and it allows us to rank the various poses. The scores that come out of binding or docking software include factors such as energetics, steric interactions, hydrogen bonding, things like that. So inputs to a docking analysis would be the structure of our chemical of interest, the structure of our protein, that usually comes out of an X-ray crystallography, and then there's certain force fields that come into play for the docking analysis around the binding site. The outputs, the 2 principal outputs from a docking analysis, are the binding poses of the candidate conformations and the affinities associated with those, essentially the docking scores. Docking has a lot of different uses, especially in pharmacology, and there's emerging usage in toxicology as well. Some of those uses are shown on this slide. So they can be used for estimating binding affinities, as I mentioned before, for virtual screening across libraries of compounds, chemical prioritization. They can be used to inform QSARs, which we've heard about previously, also predict metabolic interactions. And they're also useful in being able to predict molecular initiating events for AOPs. Shown highlighted in here or bounded in red are two of the areas that are going to be illustrated in the case study today. So there's many examples of using molecular docking in the literature, but mainly in the areas of drug design. Its use in toxicology and for environmental toxicants is growing. So this study, the case study here, which is from a paper by Kim et al., highlights the use of molecular docking in the context of chemical toxicity screening and data gap filling. It also ties in the use of AOPs, which we just heard about. And what I want to point out is that this case study is not a perfect implementation and application of molecular docking. It actually illustrates a few serious flaws that I thought would be worthwhile to cover as well. So the basis or the problem that these authors set out to solve is outlined here. Essentially, there's a lack of sufficient mechanism-informed toxicity data for organophosphate flame retardants, or OPFRs. And these compounds are increasingly used as replacements for brominated flame retardants. There were several data sets that were used as part of their study. For biological activity, they made use of Tox21 assay hit call data regarding some human nuclear receptors. There were 7 of them, and those are noted here. They also made use of data for the proteins. As I mentioned, we need crystal structures generally, and those were obtained from the protein data bank. They also used some external data for in vivo oral toxicity from EPA ToxValDB. I won't be talking about that as much in my talk today. So essentially, the authors decided to build a mechanism-based integrated screening framework to try to answer the research problem I just mentioned to you. So going into the top of their framework would be a list of OPFRs. They would look at both Tox21 assays as well as molecular docking for compounds for which assay data wasn't available. From those 2 data streams, they could evaluate the potential toxicity or bioactivity, and combining those would lead to some mechanism or some methodology for prioritizing OPFRs. Let's go into just a little bit more detail. They searched the literature broadly and identified 48 OPFRs. They then used the EPA CompTox dashboard to search for Tox21 high-throughput assays related to the nuclear receptors mentioned earlier. What they then did is they filtered those assays based on whether they referenced an adverse outcome pathway, or AOP. From this sort of screening, they found 25 assays of interest. They found that out of their 48 OPFRs, 26 had been tested as part of the assay battery of 25 assays. So that left 22 chemicals whose activity had to be predicted. So let's talk then about that prediction. What shown in the upper left here are 5 of the 9 receptors they used for docking. Those structures are based on information in the protein data bank. They also selected 2 known positive binders, and those are rotenone and bisphenol A. They ran the molecular docking simulations on those 22 OPFRs whose activities were not known, as well as for the 2 knowns, the rotenone and bisphenol A. What they did then was to say, if the OPFR's binding score indicated at least as much affinity as those known binders, they were judged to be bioactive. The thing that's notable, well, before getting to that, let me just point out that the software they used for this analysis was Autodoc Vina, which is an open source docking platform that's really widely used, and there's a lot of papers referencing it. One thing that's notable about their study is that they did not run docking analyses on the chemicals that were part of the in vitro battery. So what I'd like you to do, at least briefly, is to consider this question. Does this method of predicting molecular activity and combining with results from in vitro assays seem reasonable to you? And you can select more than one answer if you'd like. I'll just give you a minute to read over those and consider. What I would say is that responses C and D are correct. Because they required that the binding affinity was at least as great as the known binders, they may have missed some that would still bind, but with lesser affinities. So I think there's the likelihood of some false negatives. For response D, it's maybe not known to everyone out there, but most Tox21 assays are not binding assays. So it's not obvious why docking and Tox21 based bioactivities would be comparable. So let's talk a little bit then about their results. Their key results, or a couple of their key results of the research, was a workflow and a heat map that are shown here. So this workflow gave them a couple pathways to obtaining bioactivity for the OPFRs through the Tox21 assay battery and through the docking analysis. Those results then fed into this overall aggregation of the results. So in that heat map below is shown the bioactivity as a function of the OPFR, the chemical that's on the horizontal axis, and the particular receptor, which is shown on the vertical axis. So the actives are in red and the inactives are in blue. And again, they've aggregated both the in vitro and the in silico results. And at least over this battery, you see roughly perhaps little less than half of the squares in this assay heat map are highlighted as bioactive. So here's another thing to consider, a question for consideration. That is, based on the information that I provided to you and what you know about molecular docking and perhaps in vitro analyses, what do you see as some of the major strengths and weaknesses of the use of molecular docking in this study? So for screening of chemicals, as well as prioritization. I'll give you a minute to think about this. Okay, here are some of my takeaways from this particular case study. So the strengths, molecular docking, I believe, is an appropriate screening tool to identify likely binders, and it's been shown to be pretty successful both in pharma and in some studies of environmental toxicants. The use of molecular docking can also provide mechanistic support when building an AOP in terms of the molecular initiating event that then leads into the key events along the AOP. There's a couple of weaknesses that I think are significant, and one is that docking was not used on the chemicals in the in vitro battery, such that they could have examined the concordance between the in vitro activity values and those predicted through docking. And I believe testing the method with the knowns is a really critical step that was missed in the study. One other thing that we covered a bit earlier is that only a small subset of Tox21 assays are truly binding assays. So combining docking results with the in vitro Tox21 raises some questions about the validity. Overall, I would say that molecular docking is a very nice in silico method for screening chemicals, assessing binding, affinities, that kind of thing, but does, like any other in vitro or in silico method, need to be examined, and its domain of applicability and its limitations need to be considered. So thank you very much for your attention, and I'll pass the baton.
Everyone, I'm Bryant Chambers, and I'm picking up last part of this here. So I'll be covering the last little bit with a few case studies.
Bryant, sorry to interrupt, but we see your presenter view. So you just have to swap the displays.
Swap, thank you, sorry.
No worries.
Leave it to me to do this. Swap displays, where am I going to find?
Under display settings.
Oh, man.
You just had right over the top, you just had your cursor up there.
Oh, God, I'm the guy who's doing this. Where did I just have it?
Look straight up from your presentation. It's in the middle of that show taskbar, display settings, and live show.
Okay. Stop sharing. Christ. Is that working better now?
Yes.
Yes.
All right, thank you. Apologies to everybody. Hi, Bryant Chambers. I'm going to be going over the last part of this talk, which will be about AI, machine learning, and deep learning, all of which are very, very, very different topics. And we're going to look at a couple case studies here, and I'm going to try and stay a little higher level in this because of the breadth of knowledge that each of these approaches takes, and more try and talk about how these case studies and what the authors did to apply these methodologies to toxicological data so they could get a toxicological result. These models and methods have application across a host of different worlds that you guys are obviously everybody's experiencing right now. So to recap from the last set of talks here, just going to basic high level stuff. AI, machine learning, deep learning, all take various inputs. And this is actually one of the main things that we need to think about. And I'll cover that here in a second. But the general trend here is that we're looking at large training data sets. So chemical structures, assay data, omics data, multi-omics data, graph structures, images, unstructured data, meaning things like just random text chats from people all linked together in an enormous corpora, or very highly structured data where we have known classifications, known outcomes linked for very supervised approaches. Within this, we always need to be careful about how we split the data set into a test, validation, and withholded data sets. So how do we give the model enough information to learn, but keep enough of the information so that we're able to confirm that the model knows what it's doing? And lastly, how can we spy on the model to make sure we know that what it did was correct? So you've got to have different withholdings in different levels. The outputs from this can be many different types of data. So we can look at predictions for toxicological parameters. So quantitative, where we can actually compute things like values, a POD or a concentration of activity. Or we can do categorical hazard marking. Does this chemical do this? Is this something we should be worried about? We can also look at trying to classify and identify features in unstructured data sets. So even at a pathological level, you could go in and look at slides and identify features within that using AI to understand, is this weird little image of pixels that are kind of condensing together evidence of chromatin condensation, and be able to learn on all the subfeatures that do that. In a more recent period, we've started moving into synthetic generation. So not only can these things do classification and look at and defining key parameters, they may also now be able to generate data to learn or train from other models on. So it's not just that. And you've seen this now with generative AIs that can produce entire summaries for you. So this is all that's going on here. In these, there's a generalized information flow. What's shown here on the right is more of a network or a neural network type approach, where we have a structure, a bioassay. We're taking information up here at the top, and we're passing that through multiple layers that have different weights and different activities that will be fired or set the system to say, yes, I've got enough input to fire this, from a very broad level, firing into a much more specified level here at the hidden layer too. So do I have a cat ear? Do I have some kind of tail? And then in this next layer, is a cat tail plus a cat ear sufficient to say I have a cat? And can I make a prediction? Yes, I think that this is a cat. In this case, toxic, non-toxic. So I decided to go for kind of the obligatory Promethean question here, just to gauge what the audience's familiarity is with AI and machine learning, kind of just gauge that framework. So just put a word, a phrase, anything that you think. What is your feeling about AI? What comes to mind when you think about AI? And I'll give you a few moments here, and we'll see kind of where we are as a group. I love it. I do like, well, I won't say anything. I don't want to bias anything at all while people are pushing stuff through. So I'll give you another 15 seconds to come up with your feeling, kind of query yourself and see what you think. All right. About 10 seconds. All right. I'm seeing it slow down just a little bit. We're going to stop this here in a moment. But obviously, everybody you're seeing a lot of what you commonly see. I love that we've got prediction in green. And I would agree with this. There's an AI working here that's colorizing different terms. Prediction is green. It's telling us some stuff here. So we're seeing that a lot of people believe that there's some predictive power here, but we're seeing that people believe that it's undependable, or there's a lot of uncertainty in here. I would agree with the term black box. So there's a lot here as to how you use these kind of models, what you think about them, and I like that Scary's here, but also Lifesaver's here. They can be leveraged in ethical ways. They can give us lots of information, but if we're very unsure about how we're applying them, we're not careful in how we apply them to different data sets, it can definitely lead to some very uncertain or undependable outcomes. So I'll jump forward here and kind of go back through some of the stuff we've seen and kind of classify how I'm going to frame some of these models in this talk. So many models underlie AI. This term typically describes a bunch of tools that help automate complex decision-driven action, whereas machine learning tends to automate information learning as a step, like a classifier. So this is it a complex set of decisions that need to be using some kind of intelligence to get to the end of, or am I making kind of a simple assumption about is this a this or is this a that, right? None of these is wrong. And actually, many of these advanced machine learning or generative AIs, it's more just type of tool. Do you need a sledgehammer to nail a nail into the wall, or do you need a hammer, right? And what's the best way, given the type of data, the quality of data that you have? So looking at traditional statistical models and evidence-derived models, like, say, a binding model or a partitioning coefficient, these are models grounded on empirical data in a scientific framework that we're trying to wrap our minds around. You've built a model here. It's not totally true. There are many different models, like gamma-based pressure models for binding coefficients, that we don't typically use when we think about this, but are useful in, say, high-pressure wastewater treatment systems. In that situation, you need a few parameters to set up the model. You get an answer. We move a few steps forward, we get into larger random effects models where we're starting to think about, we need a lot more data to start thinking about that relative to what's necessary to solve something like a binding model. But as we move down this chain, we're starting to add complexity to the number of parameters and the way parameterization works in this system. And that's more what we have to think about is, do we have enough quality as we do that in our data? And what is the right way to solve this problem? Small tabular data is very easy to be applied to something like statistical machine learning and machine learning. So here, things like support vector machines and XGBoost, random forest, give us a lot more power in how we can go through and quickly look at that data and solve it with a lot more confidence. For some of the comments about uncertainty or unpredictability, things like random forest models or even decision trees give us an ability to look inside that model directly and see what the key parts of that model are that help the model make the decision, right? So from that, we can go into something more complicated. We're looking at multi-image data or many books and stuff like that. There, we're going to start moving out of these models where we're trying to cluster data in different spaces and assign some kind of status to it if it's in one of these spaces, and start to try and figure out more complex decision making. Do parts of this add up in a way, and how much decision is necessary to make that? And that's where we start getting into neural nets and deep neural nets. Beyond that, we get into things that look at data like corpora, entire encyclopedias all at once. And this is where we're starting to get into transformers or generative AIs. So within this framework, I'm going to pick a couple of these, just looking at something out here where we've got a case where we've got some tabular data in a complex set, where statistical machine learning is probably the better answer for it. And also something where we're looking at a larger, feature-rich, complex data set, where using a tool more like a transformer is more accurate for what we need to do. So looking at the MIE is the first part of this. And looking here, what the authors of this first case study are trying to do is be able to classify a molecular initiating event. That is, what initiating event occurred based on some type of data. Here, they're using transcriptomic data. So this is a paper by Bundy et al. Published in 2005 or 2025, a continuation of an earlier paper published in 2022. And what they're doing here is they're taking one of the main problems that we see initially is that transcriptomic data can be very easy to generate at this point. That's actually kind of funny to say, but we can now generate high throughput, complete genome sequencing data for transcriptomics, multiomics, proteomics, and so forth. Being able to interpret that data starts to become the difficult part of that. And we can do that using traditional statistical models, but then we have to mine through all of this. So if we can do that one time, learn some stuff about it, understand what those key signifiers are of that, and then train a model to go back through and be able to quickly apply itself very quickly to that, gives us an ability to quickly determine a chemical's MIE, just looking real quickly at here's the transcriptomic data coming through, and here's what we should be able to make as a best guess on this. The authors of this study are using RuffChemDB, which is a database that has a multitude of annotation streams in it to be able to understand what those molecular targets are and annotate them directly to chemicals. They also use the EPA's HTTR database, or their high throughput transcriptomic database that has tens of thousands of chemicals in it and is able to then be linked to 8 dose level responses, move that together. So a wealth of complex data able to do this. The authors immediately go into this And this is where it gets critical about how you look at that. So this is tabular data. They have an annotation of chemical activity, and they also have complex data mapped into this, which is that bioactivity data coming from the transcriptomics. Within that, they're able to use some initial filtering to look at concentration response and filter out non-responsive chemical or dose response categories. From there, they can pull a set of active transcriptomic profiles. So what they're looking at now is we're not looking at no noise, we're looking at some noise, and we've got to be able to find biological signal from that. Critically, they parse their data set into an exemplar data set. So this is kind of a small withholding data set to look at inside, plus a training data set. They pass this through, partition the data into MIE-specific training data sets. So now they're subdividing it into known areas of activity and trying to train the model. They pass this into MIE-specific classifier subsets and train the data on these known transcriptomic profiles that they've decided that have activity. One of the best things that I think comes out of this study is they use an empirical significance testing. So not only do they train this and say, yes, we got a result, they also build out fully randomized signals to say, this is garbage. Do you still give me a hit on garbage? And when does the garbage start to separate itself from the actual signal that we're trying to look for? And with this, they can filter out some of these pre-trained classifiers that they build here, and it gives them the ability to say that out of this, only 3 of the MIEs that we were looking for were we able to, out of the 9, we were able to find 3 MIEs that we were actually looking for signal on. From this, they can apply that into the large data set of the tens of thousands of chemicals, identify and assign a prediction to each one of those chemicals, look at the concentration response modeling, and then pull back to that subset of exemplar chemicals and confirm, did the model assign it to the activity that was expected? This study has a lot of strengths. The null model validation prevents false pattern detection via rigorous testing. Being able to have some kind of statistical model within this to look at how well does my classifier perform objectively is very critical to applying a model like this. On top of it, the researchers really understand what they're looking at. It's not just slap a model against data. Here, they're actually looking at 8-point concentration to ensure that we're looking at information in a dose response. Lastly, they do an external validation that we don't look at in this first image, where they're actually now, and this is slide C, I think figure 5 in this study, they look at cross-reference against the ToxCast wet lab data to look at biological credibility. And here is where you can actually see, and they're very critical of themselves in this, which is, an evidence of a very good study. When they do this, they're only getting 42% recall of chemicals that have known activity. Some of that is because they train this only on MCF7 cell lines. So this is some areas to improve. Using multiple cell lines, understanding the cell line specific context allows them to be more accurate, but they're very clear about where that comes from in this study. On top of that, this model needs at least 5 reference chemicals, limiting rare pathway analysis, which is a shortcoming. Very quickly, I'm going to go through a very interesting second case study here. So the problem here is looking at how can we identify chemical activity similar to a QSAR, the way that we're looking at chemical structural toxicity flags. But one of the problems we have here is that this can suffer from low accuracy or narrow domain of applicability. Basically, I'm looking at a very specific endpoint, and if my data doesn't directly conform to the type of structure input that I need for that or have the exact kind of framework for that domain of applicability, then it's not going to give me a solution. And I tend to lose around 80% of my chemicals to 20% of the chemicals that I'm feeding into that. Can we use something that looks at this holistically to solve the problem all at once? So this is a case where rather than using a highly focused type of ML to solve a problem, there is a necessity to use something like a transformer to do this all at once. Basically, you're keeping all the context necessary to understand all the different changes in the data all at once. Here, context provides meaning. So treating the chemical structures, the smiles, as sentences, essentially, I can say a word, I saw her duck. If you do not have enough context on that, you would understand this to be, I observed a woman lowering her head to quickly avoid something, or I noticed a duck that belongs to her. So we have to tokenize this, break it into parts, and put it into the larger context of what's happening. I saw her duck down by the river. There's still not sufficient context here. That sentence could be mapped to a multitude of things. Even if we break it up, we can get into much more complex analysis of where this sentence goes. So using what's called a bigram here, we break it into multiple parts, and then we can analyze each of these parts in context as to how it fits into the overall problem. If we looked at an entire corpus of all sentences that ever had the river in it, we could even map this sentence to Chris Farley's famous down by the river or living in a van down by the river, right? So context is what's driving this. This information, once broken into these parts, is fed into. So we've learned all the different contexts about how different versions and sentences and chemical structures work. We feed this into a deep neural net, or the researchers feed it into a deep neural net, where we're training these breakdowns or these representations of these chemical structures alongside exposure data and effect and endpoint. What this lets them do is not just say, hey, I've got a flag for this outcome, but rather, hey, here's the exact concentration that we think, or the model, meaning we, the model, thinks that this activity is going to become active for this chemical in this context. And that's the important part here, is this is a situation where understanding all of the situation at once, relative to all of the concentrations at once, is necessary to solve the context or the situation in which we want to understand what's happening here. This takes a lot of data. So here they use sources for REACH, Ecotox, and EFSA. Looking at a final trading data set of 150,000 experimentally measured effect concentrations, and they have to map this into 3 organisms. So just giving you an idea of what this looks like. What this leads to is something that's pretty cool. Integrating the EC50 and the EC10 data to capture that relationships, it's able to then understand the entirety of where is this thing going to become active. It can predict many chemical structures, unlike tools that reject out-of-domain chemicals immediately. Lastly, it learns the toxic features automatically via context models, reducing reliance on fragments. We're not looking at is a phenol present. We're saying this chemical with all of its components in the context of all of these assays will give me a response at a concentration of X. This is pretty cool. You can see that we see a mean squared error reduction of 7-fold relative to some of the existing models. This is an enormous drop, an enormous tipping point or movement forward in this kind of model. However, like we saw previously, this is a black box. We're getting numeric predictions, but we really would have to go into the model and look at which of these attenuation systems fired at one point and then we have to reanalyze that to understand what maybe drove that decision. So secondary models would have to be run to look at this to understand what happens. Whereas previously, looking at the ML we looked at, we can go back to some of those classifiers that succeeded in classifying and say, these are the drivers. It was this gene, this gene, this gene. I understand a bit more about how your decision was made. So it's kind of a balance here about what you're trying to solve. So with that, I'm going to turn it over to the panel to answer some questions. The main point that I want you to take from what I talked about today is that it's the right tool for the right problem with the right kind of data that helps us understand and solve this. So with that, I'll stop and I'll hand it back over to everyone else. And thank you very much.
Great. Thank you so much, Bryant. And thanks as well to Bridgett and also Brad in absentia. Just a note about the Q&A. There are some questions being answered in written format. So if you don't see your question, look over on the answer tab and we'll get those covered. All right, so the first question is for Bridgett. Thank you for the informative talk on QAOPs. For the example you presented on human effects, were you able to derive the main point of departure that can be used for risk assessment through this quantitative AOP?
Yeah, so that's a great question. Thank you for it. This particular QAOP example really only focused on screening and prioritization and not on risk assessment. So the majority of the key event relationships were really relating the relative responses of one key event to another. So you can make those predictions of if you had a % decrease in activity of one, what % decrease would you see in another key event?
Okay, got it.
Thank you. Let's see here. We have a question, I think, for Bryant. And this is a big question, but hopefully you can give us a brief overview. Are there any AI models which would be considered a best fit for toxicological data set analysis?
Thank you. No, that's a very good question, and it's kind of looking at, okay, how do I do this? If I'm going to start getting into try and apply some of these more large-scale models, what's the right one to apply? And the real answer is there is no perfect model. First off, everyone knows the famous there's what is it? I'm trying to remember it off the top of my head right now, but there are no good models. Some of them are better than others, right? There is no such thing as the
Useful.
perfect. What's
I think
that? Yeah.
useful is the word.
Yeah, there's no such thing as useful models. Some of them are better than others. Each of these different AI models is, you've really got to look at the kind of data, the quality of data, the kind of problem that you're trying to solve. If I'm trying to look at mapping, let's say, pathology data or digital pathology data into omics data, I'm going to first need to do feature extraction. And that's not something I can do really easily with ML-based approaches where I'm looking at classifiers. There's no tabular data there. It's not structured. The system can't learn on it. So in that situation, what you're really looking at is first to be able to learn what those features are that you don't even know. That's unsupervised learning. You need an unsupervised model to do that. And there's a world of unsupervised, unstructured learning models that are necessary to go into that. Maybe you've got that and you've got some features and you want to do some classification of outcome from features. Then you need to look at supervised models where you have known annotated feature information that you can go back in and apply to that previously applied model. The real answer is there is no best AI model, and each of these is a world. If you look at that original kind of graph that I had there, each of these is a world of different models to go into and solve. And I really think, there would be a need or a utility to maybe in one of these 101 classes really go into that and break down some of those models in detail about when is a support vector machine necessary and when does XGBoost need to start using kernel tricks to go in and do multiple clusterings. That's really complex technical jargon, but really it means that knowing which of these models is going to perform best on which type of data is probably the hardest question. The answer is, and I didn't even get to show it to you, but in the first study that I showed, Bundy and all of that group, actually there's a whole second study where they go through and they look at every single type of known model that's used in the Keras package within R and look at how does it perform for each one of these, weight them against each other, look at performance, look at the value that comes out of them. And that's what makes Joe Bundy's papers really good in this area, is that being at the EPA and doing what they do, they're really good at putting up all the evidence about why a decision was made. It's not just, hey, I ran this model and I got this great outcome, but I don't understand why. And I think that's the take home here is always be clear in how you let the model make the decision and be very careful about when you're applying a model out of its area of use. So that's what I would say there. I hope that doesn't really answer that question in the greatest way possible. More, it's like that whole, why do planes fly? It's complex. Don't worry about it. No, there's a multitude of things that we really would need to get into in this area, but mainly, no, there's not a single model that you could possibly use that is good for toxicology, but rather different data streams need different models, and you have to be very careful about what you choose there.
So I think you sort of already answered a different question that we have here about how could we define what is the right AI tool for each problem, but just see if you want to add to that.
No, and I think that there's a subtle, I think, like extension there with that, right? This gets into how are we going to regulate AI? How do we define what is a good model? When did a model work correctly? And that is hard, right? Something that was really cool this last year for me was I have to take an NIH training from time to time that tells me whether or not I'm allowed to use a computer the next week. And I always get to that last day on the last hour when I need to go to that last meeting for it. Last year, the training was AI scary, AI very worried. I don't want to see this. What is a generative AI going to do? This year, and I guess the second part of this year, it was much more positive about, this has been helping me a lot. I can review 20 papers and I have little things that I need to go back and look at. But what I'm getting at here is one of the cool things in this conversation was we were giving case studies of when was an AI used for different things and how do we analyze it and was it okay and was it ethical and was the over application of it ethical, and I think this right now is very much a caseless situation where you have to go in and look at each one of these and really think about, is this the right place where we need to be using an AI? Is it the right stage of toxicology? Is a high throughput screening area where we need the capacity of that the right place to have it? And then once we've done that screening and we get deeper into the weeds, looking at some of the data that Bridgett showed today, being able to really pinpoint, did this pathway fire the way that we need to know that it fired, and did it fire in a way that we expect that kind of regulatory system within that genetic pathway to fire is a separate part of that. And so AI isn't going to solve toxicology for us. It's going to help us streamline questions that get to our more refined approaches so that we're looking at it in the right way. And I think that's more how we're looking at AI right now in that kind of framework. And being able to go back and say which one's the best, that's going to take a lot of experts looking at things for a while now to ensure that it was done in the right way at that time. So that's what I guess that's my extension. Thank you very much, Christine. Sorry for talking ad nauseam.
No worries. We have a couple more questions. I know we're over time, so hopefully you can stay on for a couple more minutes, and we'll answer these last two remaining ones and finish up. Kyle?
Yes. So the remaining two are also about AI, the concern of the day.
Bridgett's done a really good job of answering.
Yeah.
Writing.
That's a reminder to everyone. Like Christy said, several answers are over in the answered section. I think a question that, as somebody that used to work for the EPA and deal with confidential information, a question that I have is how to maintain confidentiality and security levels when using the AI models. Is that something that comes up for you, Bryant, at NIH?
Yeah, definitely. Like, so some of this is, when do I pass something? So first off, some of these models you can run on a local machine. Some of them can be run on your phone this day and age. Right now, you're running a million things that are looking at, did you say, hey, Siri? And I'm going to watch it turn on right as I said that. And so that's happening locally, but also sometimes it's passing information to a separate entity. And the question is, what are they doing with that data? On top of it, the sheer level and the complexity of data that I need to run some of these models might entail that I have confidential data in these things. I need to look at and have lots of identifiers. If you need feature-rich data to make a health decision, I might need to know things about your age, your sex, your identity, all sorts of stuff. And I have to be really careful about how I delink that, what I feed to the model, the bias that I have in that when I feed to the model about who I think this person is, or what do I think is happening in this type of cell line, and where does that come from? And so ensuring that, so a lot of that is best practices as it is for how you store a database, what you do to encrypt a database. So I'll say that part first. The second part is understanding when a model is using external information and when it feeds that into its, and I cannot believe I'm going to say this, and don't take this too far, but it's being, meaning if it's going to use that information to adjust itself in a really meaningful way, and if I'm giving it a lot of information, am I passing information that I should be keeping to myself? And so do I need to be running a local model that doesn't give that to Google, doesn't give it to Microsoft?
Yeah.
And so right now we're working on developing some of these, the ability to run some of these things in-house, but also they're starting to get better and need less information overall to make a decision. And so that's not necessarily gonna go that distance long term. Thirdly, there's some really cool stuff happening in cryptology right now where some of these models are learning how to operate on encrypted data. So what that means is I can run a model here, take that data, encrypt it, send it to another location, and on the actual encrypted data, the model can then be run knowing how to work with that encrypted bit, send it back to me in a way that I can then decrypt it and look at what that result is. So there's some really cool stuff that's happening to make sure that that's going in a much more robust way. But really it's thinking about, are you passing information that you shouldn't be passing to something else? And I think that really kind of goes back to just best practices in general, and I'll stop there.
Okay, thanks very much. So the last question is in the hidden layers 1 and 2. I think this is going back to the original diagram. What do you expect the model to be doing?
So this is kind of just the kind of classic feed forward recurrent neural network model. This is decidedly different than something that's like kind of a cluster based model like KNNs or something like that. What we're looking at here is something that tries to start to approximate what neurons might look like when they fire. And in that situation, it tends to be the really good analogy that's been used for this historically is kind of a reverse game of telephone. Maybe you can actually play this holiday. Maybe we're creating a really cool holiday game right now. So imagine you have a game of telephone, right? And what you're doing is, you know how there's a signal decay across telephone where the game is I start with I have on a pink hat, and by the end it's I'm petting a pink cat or something. I cannot believe I made that up successfully in flight right there. But in this situation, let's say I have an image, right? And I've got many different images. The first layer of that is going to be kind of the low detail oriented layer. And that's how these things tend to work. And this is a giant generalization here and kind of an oversimplification, but it kind of mostly holds that in that layer, you're training these guys to the first set of, let's say, nodes or neurons to look for something like a little features. Do I see a blip of pixels that looks like a cat ear? Or do I see a blip of pixels? Can't remember what my two examples were at the beginning here, a pink cat and pink hat. Or do I see like a fuzzy little ball at the end of a thing? Do I see somebody's head with the outline of this, or do I see a tail, right? And so it's getting all these features. And that row of the telephone game then says to the next group, I see a tail, I see a mouth, I see some whiskers. Those next layers start to integrate that further and decide whether or not they're going to fire. And they say, I see whiskers, tail, and ears. I see I see ears, tail, and I hear a purr. That's not what it's going to do, but just play with this analogy for a moment for you. Think about all the features that I see a paw, right? And if it gets enough of that, then it fires and it tells it, maybe there's multiple layers. It really, this is, there's a lot more going on because it's not just a two-layer network sometimes. But at the end, you get to these final decision prediction fires of, if I see enough evidence of small features, that I see enough evidence of something that might look like a cat, that confirms a big picture of a cat. I'm wandering across my screen as I do this, I'm sorry. And then I get to the very end here and I say, you know what? That looks like a cat. Instead of a pink hat, this is a cat, right? And that's kind of what's happening in that. Just to use your question for 5 seconds longer and talk about how that differs from something like a transformer that is what we're using right now in these chat GPT. That's a generalized pre-trained transformer. And what's going on there is it's still very much like a neural net, but you have attention nodes, right? And what's going on in that is, instead of just saying, and I hear a common analogy that I've heard 1,000 times over, and an AI spits back at this point, if you ask it how one of these works, is it's like a detective asking people in sequence. So a neural net is, what did you see this person do on this day? And then going to the next person, what did you see this person do on this day? And then trying to integrate it all into a decision singularly. Imagine if you brought all the witnesses into one room and at one time started talking about stuff, and each witness could confirm with each of the other witnesses what they saw at one moment in time. You're getting a confirmation all instantaneously about every piece of information, rather than losing what A, B, C, D witnesses said, or one witnesses 1, 2, 3, 4 said at that. And so all of it at once is turning into this enormous, in context, full confirmatory signal. And that's how that starts to differ from something like an NN or a DNN. If that analogy is good. But maybe this holiday you have an extra bit of eggnog and you go play DNN Transformer or play neural net telephone and see what you come up with. But that's kind of, hopefully that answers that question for you.
Yeah, definitely. Okay. Well, I think we're going to close out the webinar. Thanks so much to all three of our speakers. Thanks for all the staff behind the scenes helping out with putting on all three of these webinars so far. Thanks to you for attending and listening and sharing your questions and feedback. And we hope you have a good holiday season, and we'll see you in the new year. And stay tuned for some new webinars as they are put together and announced. Thank you all. Have a wonderful rest of your week.